Good evening, everyone. Sorry, we were slightly late because of some technical issues, but we are good to go, and I welcome all of you in this live session on microbial aspects and ecological aspects of Lonar Lake. So let me welcome Professor Yogesh Shouche also, and uh, maybe we can start the session immediately because we are already 15 minutes late. So over to you, sir. Please start the session.
Yes. Okay. So thank you very much for this opportunity of giving me this opportunity of talking something about Lonar Lake. So what I'm going to talk to you today about microbes that are associated with Lonar Lake. Yesterday, Ghanekar sir must have talked about plants. So coming to microbes, when we talk about microbes first, let us understand what are microbes. Basically, microbes are the very small organisms. They are very small in size and we cannot see them with our naked eyes. But how small is small? So here you see a scale and you can see 1 millimeter over there. And microbes are in the range of size is in few microns. And just to get an idea, the 1 millimeter is divided into 1000 microns. And typically the microbes are in the range of 5 to 10 micron in size. So then you will now realize how they are. So 1000 micron makes 1 millimeter. That will give you idea about the size of the. And typically if you see the microbes, there is a range. So there are these tiny larvae and the fungi that are few microns in size. Then there are our own red blood cells which are 10 micron in size, and bacteria are typically 5 to 10 microns. Below that are viruses which are in nanometer in size. And then below that are atoms. So that will give you an idea as to what I mean by microbes. So typically when we talk about bacteria, they are in the range of 5 to 10 microns. So they are in, so microbes are in different forms. Predominant forms is bacteria, which are few microns in size. And as you can see, they have different shapes. So rods, cocci, spiral, spherical rods. And the most important property of microbes is that they are omnipresent. They are present everywhere. They are present in, almost every place where you can imagine they are present, and there is no place where they are not present. So the next in microbes is fungi, and we are very familiar with fungi. So they are multicellular. And there are yeast which are unicellular. And as I said that we are very familiar with fungi, and the most common is mushrooms that we eat, and the other one is, now the monsoon season has started, so our you will see fungi growing on the wet balls. You will see them in the bread and the rotis, growing on bread and rotis. So those are fungi. Blackish or greenish thing which you see growing on bread and rotis or even in the walls of your house, those are fungi. And we are very familiar with fungi because the bread we eat is made by yeast fermentation, and also they are involved in making of wine and beer and all other alcoholic drinks. So that way we are more familiar with fungi because we see them quite often. Then there are protozoa. We are familiar with protozoa because of malaria. Malarial disease is caused by protozoa, and also amoebic dysentery is caused by protozoa. And then the fourth category is algae. They are involved in photosynthesis, and typically in a stagnant water, the greenish color which you see in a stagnant water, that is because of algae. And finally, viruses, which are kind of parasites, they can go on their own. So they need host cell to grow, and then they grow inside the host cell. And today, everybody is aware of the viruses because of the corona epidemic that is going on. So coronavirus belongs to this category, and it is a one category of viruses that affects human beings. So these are different kind of microbes that we come across in our life. And typically bacteria are the first residents of the earth. So something like earth evolved something like 4 billion years ago, and the first primitive life form was a bacterium, which came to earth something like 3.85 to 3.9 billion years ago. So they are the oldest residents of the earth. And I'm not wrong if I say that they are ancestors for all of us. So it is believed that whatever prokaryote appeared 3.95 to 3.85 years ago, from that primitive prokaryote life, over the period of 3.85 billion years, all the living forms which we see on Earth, including human being, have evolved from these first microbe that arrived on the Earth 3.85 to 3.95 billion years ago. So that way the microbes are very important. And as I said, that they are present everywhere. So a huge microbial diversity exists on the earth. There is something like 5 into 10 raised to 31 microbial cells exist on the earth, and they are present everywhere. So they are present in the rocks, they are present in the water sediment, they are present in marine sediments, they are present in deep surfaces of the earth, they are present on our bodies, they are present on and within bodies of all the living organisms. You can imagine a place and they are there. I cannot go on listing all the places where the microbes exist. But the question is, they are so tiny. So how do we study them? Obviously, one needs a microscope to study them. And the first microscope was invented in something like 1664. It was a very primitive microscope. Robert Hooke invented the microscope, and Leeuwenhoek then used this microscope and reported what we called as animalcules. So he observed tooth scraping, water sediments, everything which he could put his hands on. He observed that under microscope and described them, what he called them as animalcules. So this was not enough to study the diversity of microbes, because just by looking at the microscope, under the microscope, you can get very little information. So people tried to develop the techniques to grow microbes in the laboratory, and around 1900, Beijering developed the techniques to grow them and study. So if you want to grow a plant at your home, what do? You get a pot, you put some soil in that, and then put a seed for the plant to grow. And then the seed germinates and the plant grows. So same way, if you want to grow a bacterium, you also need a kind of growth medium. The way you use soil for plants, you use a similar kind of growth medium for bacteria. So you grow that, get that medium. For plants, you use a pot. For bacteria, you use a dish, which is, you can see here in the picture. So in that dish, you put a medium and then put the bacteria. We use the term inoculate. You put the bacteria and then they start growing. The way seed germinates and produces plant, the bacteria also grow here and form these called as colonies. So that's how you grow the bacteria and study them in the laboratory. However, and then you use a variety of other techniques to identify them. I'm not going to the details of that. However, it was understood that time that if you use this technique, you cannot grow everything that is there. So typically what was observed was that if you look your sample under the microscope, you see 10,000 cells in 1 ml of your water, but here you can get only 100 cells in 1 ml of water when you try to grow them. What happens to remaining 9,900 was not known. So people people tried to develop some methodologies to study them, and what turned out is that when and then this fraction that could grow in the laboratory was different for different samples, but typically only 0.1% of cells could be grown in the laboratory. So people tried to develop many techniques, and finally what emerged was that instead of trying to grow them in the laboratory, if you directly look at their genetic material, and as we know, the genetic material for most of the living organisms, if not all, most of the living organisms is DNA. We have DNA, plants have DNA, bacteria have DNA. So instead of looking at bacteria, trying to grow it in the laboratory, if we directly look at the genetic material, decipher the genetic material, and try to understand that, that gives us a better understanding of what is there. And over the last, these are the techniques that were used to study the genetic material. I'll not go into the details, but the idea is that you develop techniques by which you can analyze the genetic material, understand the genetic material, and you can find out what bacteria are present in any environmental sample, including your own body. So people developed these techniques by the end of previous century. So 1990s, these techniques were discovered, and then the developments took place very rapidly. The most important development that helped us in a big way was development of faster and cheaper technologies to decipher the DNA sequence. Primarily the thrust for this for the quest for human genome sequence. So the cost for human genome sequence was exorbitant at the beginning of this century. It was 15,000 crore rupees for one genome, and time taken was 11 years. But last 20 years, the cost came down dramatically. Today, it is in the range of $100 per human genome, and the time taken is hardly a day. So that helped, and then we studied more and more microbes, and that's what has us to our understanding of microbes today. It has helped us in a long way to understand what these remaining 99.99% bacteria do. And this has a bearing on what we understand about Lonar Lake today. That's the reason I thought that I'll spend few minutes on explaining you the different technologies that are used to study the microbes. So these are technologies collectively called as next generation sequencing technologies. There are many technologies in the market, but another interesting development is that the technologies have moved far away. On one end, there is a huge machine like this, which costs several crore rupees and requires a specialized lab to study. But now you also have this miniaturized version of similar machine, which can be attached to your laptop or even to your mobile phone and take it to the field and determine the sequence of DNA from the sample. So you can now go to the field and do this kind of studies and understand the microbial content of the sample that is given to you. Now coming to Lonar Lake, we know that it is located in Buldhana district of Maharashtra. It is formed by meteor impact some 50 to 60,000 years back. It was formed by an meteor, and it is oval in shape. And the most important feature of Lonar Lake is that it has a alkaline pH. In 70s, the pH was about 12 and the salinity was also much higher. Today, the pH has dropped, but it is still above 10 and salinity is in the range of 1.2%. And so a lot of studies have been done on Lonar Lake, very important from many other point of views also. It's the only meteor crater in the basalt rock globally. It has a lot of similarities, analogies to Martian surfaces. So it is considered as an excellent analog to study the environment on the Mars also, and a lot of studies have been done on that, but I'll not be talking about that. So since it has this unique feature from microbiological point of view, that is, it has a very high alkaline pH and it has very high salt content. And in microbiology, the organisms which can grow under extreme conditions are called as extremophiles. So any extreme conditions, very high temperature, very high salt, very high pH, very low pH, very low nutrients, these are all the microbiological point of view. Lonar is multi-extremophile because it has high alkaline pH and it has high salt content. That way it is a multi-extremophile, and the organisms which microbes which could be found there are multi-extremophilic because they tolerate not only high pH but very high salt content also. So over the years, people have done a lot of studies on Lonar Lake trying to find out the microbes. And one of the simple techniques, as I mentioned earlier, was growing the microbes on the laboratory by using different culture media, which I mentioned earlier in my talk. So you studies a bunch of microbes have been described so far. Many novel or new microbes, so far 16 new microbes have been described from the Lonar Lake by various researchers, including our own research group. A large variety of new microbial species and genera has been described from Lonar Lake, that is by trying to grow them in the laboratory. People have also used DNA-based studies, which I mentioned some time back, and the studies indicate that it harbors a great variety of bacteria, fungi, viruses, and another group of aquatic organism which is called as archaea. So it harbors a huge diversity of microbes present in the Lonar Lake water as well as Lonar Lake sediment. And again, I am not going to go into the details of this, but if you look at the Lonar Lake, it's a large lake. So at different locations in the lake, the environment is different, and so the type of microbes that are present at these different locations are also different. We have done a lot of studies on this variation at different locations in the lake, and also monsoon, summer, winter, the environmental conditions differ. So we have also done studies on how the kind of microbes that are present in the Lonar Lake vary during different seasons. So those studies have also been done, and we understand a lot more about how microbes behave and function in this Lonar Lake ecosystem than what we knew about it earlier. 10 years ago, we knew very little about Lonar Lake as an ecosystem. Today, we know much better and much more about the Lonar ecosystem. And we have found some very specialized bacteria in this ecosystem. In our studies, we have looked at bacteria that produce methane in these hyperalkaline, hypersaline conditions. The kind of bacteria that produce methane, we have studied those bacteria. We have also studied the bacteria which eat methane. So we have tried to understand kind of a cycle where the organic matter that is present in the lake due to plants present nearby, that organic matter is degraded by some bacteria, made into methane, and then this methane is consumed by some other kind of bacteria. So we have kind of established this entire cycle in Lonar Lake ecosystem. There are also different kind of specialized bacteria that fix atmospheric nitrogen and also degrade the nitrogenous compound that type of bacteria are also present in a Lonar lake. So all in all, it's a kind of a complete ecosystem where all important mineral cycles go on in this environment, and there are several novel species of bacteria are present in this lake. I believe there are many more unique bacteria present in the Lonar lake which are waiting to be isolated and characterized. And we would like to do those studies, and the studies will continue. The lake assumed a very importance a couple of days back because of the lake suddenly turned pink. In this phenomena happened between 5 to 10 June. The lake turned blue, and everybody was surprised. There were many people asking different questions. Many different theories were posed as to why this lake has suddenly turned pink. But it is not something which is very unique or something very exceptional. Such things go on, happen globally. So why the water gets color? Clear water appears blue, but water gets colored mainly because of two reasons. One reason is presence of dissolved minerals. Many minerals get dissolved in water and impart color to the water. And other reason is microbial activities. And we'll talk later about the other reason. So what are the microbial activities that give color to the water? So many organisms that grow in water produce pigments, and those pigments then impart color to the water. Sometimes the live organisms give color. Sometimes when these organisms die, the pigments are released in the water and give color to the water. So one important organism that give color is red algae, Trichodesmium erythraeum. So this is a algae, larger cell size, and this organism contains pigments belonging to phycobiliproteins, which are known as, the one major pigment is phycoerythrin, and this gives red color to the water. When this alga grows, it gives red color to the water. The next one is again a smaller unicellular alga, Dunaliella salina, and this particular algae contains a pigment, beta carotene pigments, and also it contains chlorophyll, which we see in plants also, and this is the pigment involved in photosynthesis. Under the normal conditions, this alga produces chlorophyll, which is involved in photosynthesis, but under the stress, and by the stress I mean a low presence of nutrients or high salt concentration or very strong sunlight, under these conditions, it also produces pigment called as beta carotene. And we are familiar with beta carotene because beta carotene is present in other food materials like carrots. It's a predominant pigment in carrot, and which gives carrot a red or orange color. So the same pigment is produced by this algae during stress conditions, and then it becomes pink or orange in color, and obviously color is then imparted to the water where it is going, and then the water appears pink or orange or red, whatever it is. Third, and this is the structure of the beta carotene. Surprisingly, the beta carotene from Dunaliella salina is used as a food additive. It's actually available online also. Interestingly, you can see the price at the range of 4,500 rupees per kg. So this is available online in the market. Another category of organism which give our yellow color, orange color is organisms belonging to bacteria or archaea. These are smaller, few micron in size. So Halobacteria or Halobarchea, they also give this. You can see these growing beautifully. These have been isolated in my own lab by my colleague, Dr. Snehal Kulkarni from Salt Pans in Maharashtra. And these are Halobarchea. And you can see that bright pink pigmentation that you can see when they are growing in the laboratory. So they also impart beautiful colors to the water where they grow. And these are the, so as the name suggests, they are Halobacteria, Halobarchea or Halobacteria, means they are halophilic. That means they require high salt concentration to grow. So they are typically present in oceanic environments or the environments like salt pans or any lake that contains very high salt. They grow there, produce the pigment and impart the pink color to the water where they grow. So these are the different categories of organisms. When they grow in water, impart color to the water. So is it unique? Is it something unusual? The answer is no. World over, you will find many places where the water is red or pink or orange or many beautiful hues, purple, due to growth of these bacteria or algae. And I'll just show some examples of that. So these are the pigments that are produced by these Halobacteria or Halobarchea. They produce pigments of the category belonging to Bacteriorhodopsins, Bacteriorubrins. These are the pigments that are produced by these organisms. And now I'll come back to what I was saying, that is this unique? Answer is no. And what are the globally, which are the other places where you see such colored water? So this is Red Sea, which is present between Africa and Arabia, between Saudi Arabia and Egypt. This is the region which is called as Red Sea. It is part of Indian Ocean, and obviously it is called as Red Sea because it becomes red in color. In some seasons, it becomes red in color, and the studies have shown that this red color is because of the growth of Trichodesmium erythraeum. So this is the algae which grows there seasonally and gives red color to this particular part of the sea, and then it is known as Red Sea. This beautiful lake in Australia, which is called as Lake Hillier, it is perennially, it is pink. So throughout the year, you can see pink color to this lake, and this is because of Dunaliella salina and also because of growth of Halobacteria. Studies have shown that numerically, though alga is larger in size, but numerically, the algal cells are present in much lower numbers. They have a lesser pigment content, and pigment is concentrated, present in specialized globules, whereas the pigment by Halobarchea, Halobacteria is present in larger quantities. It is diffused. So if you see the overall contribution to the observed color, studies have shown that contribution of Halobacteria and Halobarchea is much more than by the algae. This is what the studies in Lake Hillier and other lakes have shown. And not only that, these lakes are also home to flamingos. And you know that flamingos have these beautiful pink colored feathers. Studies have also shown that this beautiful pink color present in the flamingo feathers is actually because of the pigment produced by this Halobacteria, Halobarchea, and they are also present in flamingos and impart the beautiful pink color to flamingo feathers. So the contribution by haloarchaeal pigmentation does not restrict to the lakes, but it also extends to the flamingo feathers, imparting beautiful pink color to. I thought this is something which I could add. So another Hutt lagoon in Austria also has the full pink and red colors, primarily because of presence of algae and bacteria. Tanzania, Natron Lake, famous Natron Lake in Tanzania, also has pink color. Sivash Salt Lake in Russia, again beautiful pink color. Then Torrevieja Lake in Spain is again known for its pink color due to algae and bacteria. Lake Coloradas in Mexico, you can see again pink color because of Halobacteria and alga. Colorado Lake in Bolivia, red color due to Halobacteria and Dinoliela, Redba Lake in Senegal. See, some of the lakes, these lakes have been thoroughly studied in microbiologically. Like Australian lakes have been thoroughly studied microbiologically. A lot of organisms have been isolated. Some lakes, the studies have not been done and only speculate that their pink color is presumably because of Halobacteria and algae. So Redba Lake in Senegal. Tunisia, again, these red lakes are known, the microbiological, they have been studied. And again, it is known that this red color is because of Halobacteria, Halobarchea. It is reported in the literature. And then these grand prismatic springs in Yellowstone National Park in USA, you can see beautifully that multicolored hues. They are blue at the center, and as you move on towards the periphery, it is yellow and orange and green. It is green and yellow and orange. This is because there is a variation of temperature from center to the periphery. There is a variation in the temperature. Center is very hot, and as you move towards periphery, it goes on becoming cooler. So nothing grows in the center because the temperature is very, very high. So bacteria grow in the center. Of course, there are exceptions, but the pigment producing bacteria cannot grow in the center. But as you move towards periphery, the temperature gets cooler and the bacteria grow, and then different bacteria grow at different temperatures, and then the pigments produced by them are different. So something which is growing here produces green colored pigment, something which is growing here produces yellow colored pigment, here produces orange colored pigments. So different type of bacteria producing different colors gives these different color hues to these beautiful grand prismatic springs in USA. So in summary, there are many bacteria present in these another point to note is that grand prismatic spring is not hypersaline, hyperalkaline. pH is towards neutral. The salt content is also towards normal. However, it is still extremophilic because it is hot spring. So the temperatures are very high, above 87, between 85 to 95, between 67 to 95 degrees. So here it is 95, and as you move towards the edge, it is 67 degrees centigrade, and in the between there is a range. So it is extremophilic from that point of view, that it has a very high temperature. So the having colored water is something not unique. It is observed globally. There are many water reservoirs across the world which show different colors because of the growth of different kind of bacteria. So what about Lonar? Why Lonar would have achieved this pink color? So it is obvious, and it is by and large foregone conclusion that this pink color is due to microbial activity. Definitive answer can be obtained only after we get the samples and we actually study them microbiologically. But from whatever we have seen is happening globally, and even looking at the hue of the color, one can safely say that this pink color could be because of, one, obviously Dunaliella, and secondly, Halobacteria or Halobarchea growing there. And what is the proof of this? So our own studies have shown that the Lonar Lake does contain archaea. It does contain archaea belonging to Halobarchea group, which have shown to impart pink color to the water in other environments world over. So there is a reason to believe that under certain conditions, and here the conditions could be intensity of sunlight or the strong heat evaporated the water, salt became more concentrated in the lake, and that kind of forced these bacteria as well as algae to produce more pigment, and the lake acquired this pink color. And once either the intensity of the light went down, or because of the rain or whatever reason, the water got diluted, then the pink color, the pigment production was reduced, and the leaving pigmentation reduced. So there is a reason to believe that this phenomena would have occurred in the Lonar Lake because as I'll repeat it again that our studies have already indicated the presence of organisms belonging to Halobarchea group in the Lonar Lake. It is always present, but under the extreme conditions, their number increases, the pigment produced by them increases, and thus the lake becomes pink in the color. So this is what we believe would have happened in the Lonar Lake. And the study which I have presented here is our several years work. Many of my students, Aijaz Vani, Venkat Prasad, Anthony, Deepak, Satish and Dheeraj, they have contributed to this work, and collaborations with Dr. Ranade, Colin Murrell and Christoph Tebe have led to these studies and these conclusions. Thanks.
Thank you, sir. Really very nice lecture. So just give me 2 minutes. There are some questions. So I also request all the participants who are listening to the lecture live, if they have any questions, they can post in the live chat window in the YouTube. So there are a few questions, sir. So first question was the pink color of Lonar Lake is mainly because of the Halobacteria. I think you answered this question.
Right, yeah.
So then the second question was, is Halobacteria produces in unfavorable condition?
Halobacteria, can you repeat the question?
Halobacteria are produced in unfavorable conditions. Is it true?
Halobacteria are not present in unfavorable conditions. They are in any environment, they are always there. So they are a normal habitants of such hypersaline environments, not in unfavorable condition or something like that.
Then one question is regarding the Yellowstone National Park photograph that you showed. So question is why there is variation in temperature in the lake?
Because the hot spring geyser, I mean, this is what I understand, but I cannot say that I'm 100% true, but this is the reason is because the hot water geyser, I mean, the origin of the hot water coming from the below the surface of the earth is in the center. So that's why the water is hottest in the center, and then the water, as it spreads in the lake, it cools down. So towards the end, it starts getting cooler.
Great. Let me check if there are any more questions. Okay. So someone is asking where we get information about microbes in detail, like any reference book or something.
I mean, there are many basic books of microbiology. There are many standard textbooks in microbiology. I mean, when we were students, we used to study books like Stanier or Microbiology by Pelzer and Reed. So these standard books, Indian editions, are available. But nowadays, many faculty members in many colleges, including the people in Pune and Ahmednagar and Solapur, the faculty members have written textbooks in microbiology. So many, a lot of such basic information is available in those textbooks. But in today's scenario, really doesn't have to go to the textbooks also. A lot of information is available on the internet also about the microbes. So websites of many universities and organizations like American Society for Microbiology, their websites also give a lot of information about microbes.
Great.
So you have to just look for what kind of information you are looking for, and you'll get that information.
Sure. Okay. question is, algae are survive at any temperature? Like,
algae normally, there are some algae which survive at higher temperatures, but the normal routine growth temperature of algae is the room temperature.
okay.
Ambient temperature. I would not say room, the ambient temperature.
Okay. Then there is one question. Thermophilic cyanobacteria can survive at higher temperatures?
Right, yes. Thermophilic cyanobacteria can survive at higher temperatures, yes.
Then one question is, what happens to halobacteria when there is favorable condition?
Favorable condition in terms of what? See, halobacteria grow under high salt conditions. So if whenever there is a high salt in the environment, they will grow and they will proliferate. If the salinity goes down in any ecosystem, then obviously halobacteria, the strict halophilic bacteria, will not be able to grow if there is a very high salt concentration.
So can we take a few more questions, one or two?
Yeah, sure.
Then there is one question. Has this phenomenon, like the pink color of the Lonar lake, is observed in the past? Are there any records?
The best of my knowledge, it was not recorded having a pink. Informally, when this phenomena happened, when I was talking to different people, in conversation, some person told me that this is being observed for last 1 or 2 years. This is being observed. The coloration was not so prominent, but the pinkish color was seen earlier also for the last 1 or 2 years. This is what somebody told me, but it never became a news earlier.
Okay. There is one question. Are halobacteria also used for commercial purpose since they produce red pigments?
Halobacteria are used for commercial purpose. The pigment produced by halobacteria, again, I don't think it is commercially, but there are certain other products of halobacteria which are being commercially used. And for example, in order to survive in high salt conditions, halobacteria produce certain compounds, and one such compound is ectoine, which is responsible for their osmotolerance, and this ectoine is used in moisturizers. So there are some other products of halobacteria also which are used commercially. But ectoine is the example which could come to my mind immediately.
Okay. Someone has asked 2 questions. Why these pigments are produced by the microbes? What role do they serve? And the second question is, you mentioned the production of pigments are induced by stress. Can you please elaborate on that?
Okay. So the pigment, there are different reasons for producing the pigment. The one reason is that they are involved in photosynthesis. So the chlorophyll category of pigments and phycoerythrin and all, these are involved in photosynthesis. They capture the sunlight, and then the energy of sunlight is then used for fixing the atmosphere with carbon dioxide. So that is one role for this pigment, light harvesting. And the second reason for the beta carotene and other this kind of pigments is that they are produced under the stress condition, because they help organism to survive stress. And that's the reason we say that beta carotene is vitamin A, and that is the reason vitamin A is also useful to the body. So these are some of the known functions of the pigments.
One more question. Temperature is always high in the region of Buldana during summer. So need to know what exactly happened at this period of time. Maybe this year.
See, this is something, I agree that this is something which we have been observing this year and probably for last 1 or 2 years, if informal discussions are to be believed. And we really need to look into this phenomena. I really don't know what could be the reason. One reason could be earlier there was some seepage of groundwater. There were some other streams coming into the Lonar. Probably they have stopped this because of the conservation activities. Those inlets have stopped. The dilution of the water has stopped. That could be one reason, but one really needs to study. It is often difficult to say what could be the reason why this is happening for last 2-3 years. We have to study that.
Let me check if there are any more questions. Okay, someone mentioned that in 1970s, pH level was 12 of the Lonar Lake.
Right.
But in that time, pinkish algae didn't grow there.
We don't know. I mean, see, those type records were not available. If somebody had some notice and these things, probably it was not noticed. But again, as I said, that why it is happening nowadays, we have to really study that. It's difficult to make any predictions.
I think we can just take one last question.
Right.
In multicolor lake, different colors are observed due to different microbes. Is it a temperature effect growth of the microbes? And
it is the reason for that is at different temperatures. See, for all the microbes, there is something called as optimum or good temperature for the growth. So for some it is 20, some it is 25, some it is 37, some it is 40, some it is 45. So since there is a temperature gradient from the center to the periphery of the lake, at different temperatures are suitable for the growth of different type of bacteria, and thus number of that type of bacteria is higher at that particular temperature, and that's why those bacteria grow and then they produce a specific color pigment. So that's the reason different bacteria grow and produce different color pigments.
I think it's already 7:30, so we may stop. There are no more questions as such. So we are good to stop. Thank you very much, sir, for this wonderful lecture. It was really nice and really a lot of information, in fact.
Thank you very much. Thank you. Thank you. Pleasure. Bye.
Thank you. Bye. So we are just closing the session.
Yeah.
Thank you, sir.
Thank you.